Welcome, my students. Today we will investigating the effect of temperature on the enzyme activity. Our enzyme that we're going to use today is trypsin enzyme. Trypsin is an intestinal secretion, okay, and work on the protein in order to break it down into amino acids. Today our protein sample will be the casein. Casein is the protein found in the milk. Regularly, casein is being broken down by the enzyme trypsin in order to be broken down or metabolized, catabolized in particular, into amino acid, basic amino acid. And this actually a little bit slower procedure which gives a prolonged supply of amino acid to the body, or constant supply of amino acid to the body. Today our sample will be containing the powdered milk. The powdered milk containing 3% of the casein, and the enzyme that we will use is trypsin. Here we have the enzyme trypsin. Okay, the enzyme trypsin that we will be using today is on a concentration of 3%. Okay, 3%. Here we have the powdered milk that have 3% of the casein. Here we have to maintain the pH optimal because actually it is not the dependent variable. It must be kept constant, not to affect the enzyme activity. And here we have the buffer solution in order to maintain optimal pH. Okay? The independent variable is the temperature. So we will use a wide range of temperatures, vary from the room temperature, which is 20 degrees Celsius, then 40, 50, 60, 70 and 100 degrees Celsius. So we have to have six tubes. Each one of them will be labeled with a marker X. I'll let you know why in a couple of seconds. After that, I have to add 2 centimeter cubed of the enzyme trypsin, the 3% enzyme trypsin, and in addition to it, I will add 2 centimeter cubed of the buffer solution. In another test tube, I will add 10 centimeter of the powdered milk that do contain the casein. I have to do this for consecutive 6 test tubes. Okay. Before starting the experiment, I have to place those 2 test tubes in the same temperature that I will work on in order to equilibrate the temperature, in order to be sure that each one of them has the same temperature or starting point of temperature. Okay? For having a control, here we have 10 centimeters of the casein milk without the buffer solution, without the enzyme. It will be used for control, for better comparison. In order to prove that, the enzyme is needed to break down the protein casein. Here we have another test tube that have 2 centimeter of distilled water and 2 centimeter of the buffer. Has no milk, which has no protein in this case, and has no enzyme. Also will be used as a control in order to be sure or to prove that enzyme, and which is the main biological catalyst, and the substrate, which is the casein found in the milk. Okay, in this case we have a powdered milk needed for the reaction to start. So those will be used as a control. The casein with no enzyme, okay, to prove that enzyme needed, and another test tube that has the distilled water instead of the enzyme and the buffer solution, but lack of the powdered milk, which is the substrate. Yes. So how can we run the procedure? Safety has to be extremely taken care, because we are dealing with a wide range of temperature by 100 degrees Celsius in order to avoid the spoilage and avoid the burn as well. Okay? So we have to place both test tubes in a water bath. Here we have the water bath. But we have many techniques in order to deal with the water bath. One of them is to use a thermostatically controlled water bath, adjust the temperature, place the 2 test tubes here in the mobile rack in order to be sure that the temperature adjusted properly. I prefer this actually, but it's really time consuming. It will take a lot of time. But now we can do the opposite way, which is here I adjust the temperature to 100 degrees C. So this is the maximum temperature. Okay, how can I do this? The second option, which is a little bit faster in my opinion. We will do the following. Before even using the test tubes, I will use this, okay, hot water, okay, labeled beaker in order to take the 100 degrees C water. And I will place it in the test tubes. And I will use the water, the tap water, for only one purpose, for decreasing the temperature. So if I decrease the temperature from 100, so I can adjust where I want the temperature to be, okay, ready. So for example, I will take here some of water, sufficient enough to cover the test tube. I have to take care of that hot water. Afterwards, here is the tap water. How can I adjust the temperature? I will use a thermostat, okay, or a thermometer, sorry. Here is temperature is the room temperature, which is 20, okay. Just to prove, okay, the procedure that will be used. Here is the temperature of the water has to be, or it is 100, according to the reading of the thermostatically controlled water bath. So the following will be, I will start from the 100 downward. If I want to adjust the temperature of the water bath, I will add the water to decrease it from 100 to 80, using or measure the temperature by the thermometer. Then I will add another amount of water for another run, which will be up to 70. If I add another amount of water, temperature will be decreased to 60, okay, 50, 40, 20. You got the point? Then I can make more than one run at the same time, okay? Each one has to be for about 20 minutes. You have to wait for 20 minutes intervals. So let's get started. Here the temperature is 100 degrees C. I have to add tap water, okay? Yes. Then I will use the thermometer. It will be hot, I'm sure, because I added, now it jumped up, rocketed to, 40 plus. Yeah. Actually, I will decrease a little bit, and I will add more of the tap water. Yes, now it's, if you close, now it's about 30. I add another amount of water. Yes. Now it's become even less. Okay. I will use advantage of time in order to present it as a demonstration. Afterward, each one of you can do this in groups. Okay. Now it is, still, because I started by 100, okay, myself, I'll throw all of it and I'll just take a tap water, which is by default at room temperature, and I will measure it as well. Yeah, it's a dropped. Yes, now it is 20. So I will take the 2 centimeter, okay, cubed of the buffer solution. You have to use the pipette, and be sure not to mix the pipettes in order to avoid the contamination, and that will lead to the wrong results. Then I took 2 centimeters cubed of the enzyme trypsin. Then I will add it into the water bath, okay. And here I have 10 centimeter cubed of the powdered milk, 3% casein. I have to keep it in this water bath for about 10 minutes to equilibrate the temperature, to be sure that the temperature of the milk and the temperature of the enzyme are equal, have the same starting point. Okay? Then I will mix the casein with the enzyme and the buffer, and I will give them about 20 minutes in the water bath. During those 20 minutes, I will agitate and mix them thoroughly in order to be sure that they are reacting. Okay, where is the end point? Who can tell me? Where is the end point? We have, if you look here, the milk is cloudy, is murky. It is murky, it is cloudy, it is turbid, because it has the protein casein. Okay, so the enzyme work on the protein casein and break it down into amino acids. When the amino acids are broken, they will be water soluble. They will be soluble. So the clarity, exactly. Exactly. The solution will be clear. My question, how can I identify that? Here I have the X. On a white cardboard, or even in your lab coat, okay, if you can clearly see the X, okay, that we labeled, okay, on the test tube, it means that the protein casein is being broken down by the enzyme trypsin, and the solution is clear. Actually, I will use the word clearer, because it will not be very clear. Okay, reason. What is the reason? Yes, exactly. In addition to that protein, there are fats. So the fat will also play a role in the turbidity, okay, to make the solution unclear or cloudy, murky. So I have also another point in order to tell you here as a trick while using it. You can rotate, okay, from time to time. Me myself, I'm gonna say after 3 minutes intervals, okay, among the 20 minutes total in each water bath, okay. When you're rotating the test tube, a small, okay, clots will be there. It is indication for the starting of the breaking down of the casein milk into amino acids. Okay, so also you can use it as a point of determination of the starting of the reaction. Okay, yes. So now I'm gonna mix both. I will measure 10 minutes using the So after 10 minutes, I will add the casein to the enzyme and the buffer solution. Now actually I can't see clearly the X or the label X. I will leave it, okay, for 20 minutes. This is a 20 degrees Celsius distilled tap water. And as I told you, I have to thoroughly mix them in order to be sure that the reaction takes place. I have to leave it for 20 minutes. I'll use a stopwatch. Our end point is when you can see the X. The faster seeing the X, okay, it is the fastest enzyme activity. Okay? What do you think that 20 will do? Will it be faster, slower? Slower. Yeah, exactly. But there will be reaction. Yeah. Why it will be slower? Yeah, exactly. The temperature is lower, so the kinetic energy will be less. So the number of successive collisions between the substrate, which is the protein casein, and the enzyme trypsin, will be less. So less frequent they will be colloid, so that less formation of enzyme substrate complex, so less products. Exactly. Okay. We still need to wait for about 20 minutes, and every 3 minutes thoroughly mix them. After 20 minutes, okay, I actually noticed that the end point here is after 20 minutes. Yeah, and it's very, very, very small clotting, which is indicating that the enzyme started. It's a very slow reaction because actually the temperature is low. It is 20. It is not the optimal temperature for the enzyme to work. Now I'm going to move into the other temperature. Now I will do the rest of the different temperatures. In order to do so, actually I may use the thermostatically controlled water bath and starting from 20 and raise it up to 30, 40, 50, 60, 70 and 100. But as I told you, it takes a lot of time, and I have a class after less than 10 minutes. So I will use a different procedure in order to clearly give you this demonstration, which is I'll start by the highest temperature, and then I will decrease the temperature because it's easier for me to handle it outside of the thermostatically controlled water bath. I can have even a Bunsen burner and separate Bunsen burners, and each one has a separate beaker with a temperature that has to be maintained, which is perfect as well. But here I'm gonna use a hot water at 100 degrees Celsius. Yeah, it's rocketed now. It is now, yeah. And even to make it much more conventional, because as you know, the denaturation will be above, closer to 50 or 60. So I will use it at 80. Now it is 80. So I will mix the 10 centimeter cubed casein with 2 centimeter cubed enzyme and 2 centimeter cubed of the buffer solution. I will thoroughly mix it first. Here is the X, and I place it here. At the same time, I can make use of time. I make another run here for 60 degrees Celsius. I have to actually take care. If you bring me a thermometer, please, I'll be thankful. Thank you. This is room temperature. Yeah, it is 20. I have to have this. It is, of course, more than yeah. So here, yes, I have to adjust it to 60. Now it's 60, exact 60. Okay, so this is the third run. 10 centimeter cubed of casein with 2 centimeter cubed trypsin enzyme and 2 centimeter cubed of buffer. I will thoroughly mix it. Okay, here I will make the temperature 40. Okay, I need another thermometer, please. Okay, I will be sure that cold water is ready. I need a tap water. Why? Because here the temperature is really, really high. I have to decrease it to the optimum or the temperature needed. So I need thermometer, please. Thank you. It is 20, which is room temperature. So I have now 3 different water baths. This is 80, 60, and here is, yes, I have to decrease it a little bit. Okay. Still high. It's still high. I will use the tap water. Yes. Yeah, now it is decreasing. Still above, between 40 and 50. I need it exact 40. $45. Yeah, now it is 40. Okay. Then those that have in the stopwatch for each one, as I told you. Okay, the third one, please. 10 centimeter cubed of the powdered milk with 2 centimeter cubed of the enzyme and 2 centimeter cubed of the buffer in 40 degree. I will mix them thoroughly. Okay, here I will make it 30. I have to add small amount of the hot water, just small amount. Then I need a thermometer, please. Yeah, each one, please, with his own. Yeah, also for the stopwatches as well. Here is the hot water, tap water. I need more tap water. We still have little bit time. Here is a 20. Yes, please. Again, this is the 30 degrees Celsius. Okay, I will add the 10 centimeter cubed of the powdered milk with 2 centimeter cubed of the enzyme and 2 centimeter cubed of the buffer solution. Yes. Here is 80 degrees Celsius. No reaction. Here is the 60 degrees Celsius. No reaction yet. Here is the 40 degrees Celsius. Yeah, it just started. Okay, we still have time. Here is 30. Just put it. Okay. Each one, please, with its own stopwatch. Okay, thank you. Perfect. Here is the end point that we got in the water bath for the temperature 20, which is the room temperature. Okay, the reaction occurred, but actually it took about 19 minutes. It took a lot of time for the X to be shown, or the turbidity, okay, to be clearer. And if you have a look here for the temperature, the water bath at temperature 30, we're gonna find out that the chemical reaction occurred, but it takes less time. Okay, about as we noted, it took about 14 minutes for the chemical reaction to occur. It is still below the optimal temperature. Here is temperature 40, and we can see here there's a huge difference between this the reaction in the temperature 40 and the reaction at the temperature, for example, 60. Here it is totally unclear because actually the 60-degree temperature causes the enzyme to be denatured. So the enzyme is no longer effective, so no reaction will be occurred. But here it's easily seen. You can see the X, okay, if I have both of them against my lab coat, you can actually know the big difference between the reaction that takes place in an optimal temperature, which is 40, and the reaction that takes place in the temperature which is above the optimal, which is in this case 80. 60, I'm sorry. It will be the same thing in case of the temperature 80, because also the enzyme will be denatured. Okay, so here we investigated the effect of the temperature on the enzyme activity, and we noticed that at room temperature, the enzyme is very, very, very slow working because the less kinetic energy, so the enzyme and the substrate, in this case the casein, is not frequently meet together. But when we raise the temperature, so gaining kinetic energy, so the enzyme and the substrate are little bit meet or bounce or colloid, so we have more number of successive collisions, so the action started, and it takes longer time, but it started. But at the optimal temperature, we actually could see obviously that the enzyme worked proper or worked the best at the 40-degree temperature, and the amount of the kinetic energy are sufficient for the enzyme and the substrate to meet each other, so the number of successive collision is really more, and the reaction occurred, and the turbidity declare, and the solution become a little bit clearer. It is not very clear because we still have fat droplets. But if you compare it to the 60-degree temperature, you're gonna find out that the enzyme is totally denatured and is no longer effective, and there is no reaction occurred. Of course, will be the same thing with 80, and even if we use it in the 80 degrees Celsius in the thermostatically controlled water bath, will be the same thing. Now to turn to do the same experiment and to record the results and plot in a graph between the temperature and the time taken for the solution to become clearer. Okay, thank you. Take your time.